Hello, my name is Kathryn Atherton, and I am a student in the Bioinformatics graduate program at Boston University. Today, I am going to be talking about my group's work entitled Genome-Wide Association Study of Chronic Traumatic Encephalopathy, or CTE. CTE is a neurodegenerative disease defined by deposition of paravascular hyperphosphorylated tau, or p-tau, in the sulci of the brain. You can see the stained paravascular p-tau lesions in multiple brain slices in Figure 1. Currently, CTE can only be diagnosed by assessing the p-tau burden in the brain post-mortem. The major risk factor for CTE is repetitive head impacts, or RHI. However, people with the same RHI exposure develop varying severities of CTE, and some don't even develop CTE at all. This suggests that other factors, including genetics, might play a role in this disease. To test whether genetics alter one's risk of developing CTE, we performed the first genome-wide association study of CTE with the world's largest brain bank of CTE cases with matching controls. It is important to note that because this is human data, collecting controls that match the cases in terms of contact sports played and duration of play is difficult. In this data set, we have 237 subjects with known exposure to RHI in the form of contact and collision sports and/or military service. Of the 237 total subjects, 144 were adjudicated to have CTE. All of these subjects were genotyped for over 7 million SNPs, and their allele dosage for each SNP was computed. Additionally, we measured the tau burden in their brains both quantitatively, based on digital slide scanning, and semi-quantitatively, based on neuropathologist assessment across multiple brain regions commonly affected in CTE. For the quantitative tau measurements, a computer algorithm analyzed digital images of brain slices stained for p-tau and calculated the density of p-tau in various brain regions. For the semi-quantitative measurement of tau burden, a neuropathologist assigned a level of severity from 0 to 3, 0 being no tau burden and 3 being the most severe tau burden in various brain regions. Imputation was done on some of the low-quality samples of tau burden using mice, an open-source R package that uses a predictive mean matching imputation method. We used logistic and linear regressions to analyze the relationship between the various CTE-related traits as the outcome, and SNP dosage with age of death, sex, and 10 principal components of population substructure as covariates. Additional models also adjusted for years of RHI and/or stratified by self-reported race. After running all models as described, we found 3 significant genetic loci which had a lambda value for the QQ plot less than 1.05, a minor allele frequency greater than 0.05, and a SNP p-value less than 5 × 10 to the negative 8. These results are shown in figures 2 through 4. The significant locus at chromosome 6, shown in figure 2, is associated with CTE stage outcome and has a minor allele frequency of 0.11. This locus is upstream of the gene ASDN, which is associated with cell adhesion, the blood-brain barrier, and the immune cell transmigration pathway. Similarly, the significant locus on chromosome 5, shown in figure 3, is associated with tau burden measured semi-quantitatively in the superior temporal gyrus and is downstream of the gene CDH9. This locus has a minor allele frequency of 0.38. CDH9 is shown to be associated with various cognitive functionalities, such as educational attainment and risk-taking behavior. It is expressed in the hippocampus, the amygdala, and the frontal cortex, 3 brain regions that are significantly affected by CTE. Finally, the significant locus on chromosome 21, shown in figure 4, is associated with tau burden measured semi-quantitatively in CA4, a region of the hippocampus. The locus has a minor allele frequency of 0.48 and is upstream of the gene VN2R20P, a pseudogene that has been associated with Alzheimer's disease, another neurodegenerative disease that is similar to CTE. These results indicate that there are genetic factors that may impact the normal functioning of the brain and lead to progression of CTE. The candidate genes identified may implicate disease mechanisms, provide targets for therapies, and guide counseling of athletes regarding risk of play. This is the first CTE GWAS performed yet. However, the sample size is small. As such, efforts to recruit more brain donors are underway, and the study will be replicated. Additionally, the candidate genes found in this study will be further investigated with an independent CTE data set in the future. Thank you for your time. Please come to my poster chat session if you have any questions.